Good morning to all of you students of BT first sem. Today, I will discuss about an important chapter in your cell biology, that is your lysosomes. After this, we will do about the peroxisomes and other things which are related with the lysosomes. Okay, so let's get started now. So, we will discuss about lysosomes today. Now, what are lysosomes actually? Before lysosomes actually was discovered in a unique way. So, during electron microscopic studies, uh, scientists have found some dense body, some rounded dense body they have observed within the liver cells. And these bodies were initially discovered by the scientists. They don't know the function actually. So they described that dense body within the cell called as uh, pericanalicular dense bodies. So I'm writing the name pericanalicular dense bodies. Okay. So before lysosomes was discovered, when lysosome was discovered, they were discovered in the within the liver cells actually initially as a round dense bodies, and scientists have named that dense bodies as pericanalicular dense bodies. And such a term, pericanalicular dense bodies, is identifying as their location of the lysosomes, not the function. So the function of lysosomes was not actually discovered at that time. Then in 1955, in the year 1955, a scientist known as C.D. Dewey, C.D. Dewey, have discovered these lysosomes again based on the function of lysosome, because C.D. Dewey have I find it out that these pericanalicular dense bodies, what we call as today as lysosomes, these dense bodies contain some enzymes. They contain some enzymes that has the capability to do the lysis, to break down the cell, to break down the components of macromolecules. So renaming of these pericanalicular dense bodies has been done by C.D. Dewey and name it as lysosome. Okay, lysosome means they contain some digestive enzymes which are capable of cell lysis. So a lysosome is therefore called as lytic body. It is called as lytic body. Okay. So this was the discovery of lysosome. In this way, lysosome was discovered. Now, if I say about the structure of lysosome, if I say about the structure of lysosome, structure of lysosome, lysosome appear as dense bodies in the cell and are surrounded by membrane. So lysosomes, they appear as dense bodies, as I have said, within the cell, and lysosomes are also surrounded by a membrane. Okay, it is surrounded by a membrane. It is surrounded by a membrane, and interior of the lysosome consists of a granulated stroma and a vacuole. Interior sorry for that. Interior consists of stroma and vacuole. Okay, vacuole. Now the shape and the size, the density of the lysosome, they vary greatly. It has been observed that the size of the lysosome can be of your 0.23 0.23 micrometer to 0.50 micrometer in size. So the size of the lysosome can vary between 0.23 micrometer to zero point 0.5 micrometer in size. So due to this variability of the size of the lysosome, identification of lysosome is rather very difficult. What we know as lysosome is the presence of lytic enzymes. Various type of lytic enzymes are found. Lytic enzymes are found, right? And although the lysosomes are believed to be common in animal cells only, distribution of lysosomes in the animal cells also suggests that they may be found in the plant cells also. So lysosomes can be also observed in the plant cells. It can be found in neurospora as well, which was discovered by P. Mantel in the year 1964. So in the year 1964, your P. Mantel have discovered the presence of lysosomes in neurospora. Okay, so Pimentel in the year 1964 have discovered lysosomes in your neurospora. Okay, now various type of enzymes are found within the lysosomes, which we will discuss now. Now each lysosome consists of about 40 types of hydrolytic enzymes. So lysosome contain about 40 types of lysosomic enzyme, hydrolytic enzymes like protease, nuclease, glycosidase, lipase, phospholipase, phosphatase, sulfatase. So I'm writing the name. So it uh, wait for a minute. So it consists of hydro 40 types of hydrolytic enzymes, and the most common enzymes which are found as protease, then nuclease, then your glycosidase, then your lipase, okay, then your phospholipase, then your phosphatase. So different type of hydrolytic enzymes are found which are enough to carry out the cell carry out the degradation process of macromolecules. Then your sulfatase, right? Now the activity of these hydrolytic enzymes require acidic pH, right? So for the activity of these acid hydrolases, lysosomes have to maintain a pH. So the pH of the lysosome inside the cell, the pH of the lysosome inside the cell will be around 5 inside the lysosome, and outside the lysosome, that is on the cytoplasm, the pH is around 7.2, which we know. So you can see that inside the lysosome they have to maintain the acidic pH for the activity of these all these type of enzymes, hydrolytic enzymes. Now somehow if these enzymes leak into the cytosol, they can do little damage at high pH there, because outside the lysosome the pH is quite higher, which reduces the activity of these lysosome enzymes. So little damage will can it can cause outside the lysosome. Okay, now to maintain the pH, obviously there must be some transmembrane proteins around the membrane of the lysosome. And wait for a minute, what happened, let us see. Okay, so to maintain the pH of the lysosome, there must be some transmembrane proteins which helps to transport the final products of digestion to the cytosol. Okay, and it also has proton pumps also, like the proton pumps which we have discovered during active transport to maintain the H plus ions inside the lysosome. So lysosomal membrane, as we have seen, that lysosomal membranes contain transmembrane proteins. So I'm writing it contains transmembrane proteins. Okay. Now, what is the function of transmembrane proteins? These are found on the surface, on the membrane of the lysosome. Now, these transmembrane proteins helps to transport the final products of digestion in the cytosol. Okay, so lysosomal transmembrane proteins, what they do after degradation, after digestion of the macromolecules inside the lysosome, these products, the final products of digestion, will be transported to the cytosol. Now there they will be either excreted out or reutilized by our cell. So the transmembrane proteins helps it helps to transport it helps to transport the digested products digested products to the cytosol, to the cytosol. Okay, where further they will be excreted or reutilized by the cell, where these products will be excreted or you reutilized by the cell. Okay, now lysosomal membrane also have some proton pumps. They also have some proton pumps which will utilize the energy released due to the ATP hydrolysis, and will these pumps will, with the help of ATP, they will pump the protons, they will pump the H plus ion into the lysosome, and thus maintaining the lumen, that is the interior empty portion of the lysosome. Acidic pH has to be maintained inside there of about 5. So if I draw a diagram of lysosome, it will be look like this. So I am drawing the diagram. So this is your lysosome. Okay, the size of the lysosome is about, if I say in millimeter, it will be of 0.05 to zero point 0.5 millimeter, or if I say in the sense of micrometer, the size of lysosome is 0.23 up to, it can be of 0.50 micrometer in size. And inside the lysosome there is acid hydrolysis, there is acid hydrolysis. Okay, different type of, yes, acid hydrolase enzymes are there, like nuclease, protease, glycosylase, lipase, phosphatase, sulfatase, phospholipase. Okay, now inside the pH has to be maintained of around 5. I am writing equal to, but you should write around 5, approximately 5 degrees Celsius. The pH is there, and they are maintained by certain pumps. They are maintained by certain pumps, proton pumps, with the help of ATP. Suppose the proton pump is right here. I'm drawing with green. These proton pumps, what they do, they utilize the energy. They utilize the energy. So they are hydrolyzing the molecules of ATP to give out ADP and inorganic phosphate, and by that time it is, what they are doing, they are promoting the entry of the H plus ions, they are promoting the entry of the protons, right, inside the cell, inside the lysosome, sorry. Okay, now outside, so if this is the cytosol, outside the pH is quite higher, pH is maintained around 7.2 there. Okay, and inside the pH is around 5, as I have mentioned. So lysosome consists of number of acid hydrolase enzyme which are active only at acidic pH, and that's why we need this type of proton pumps. Okay, the proton pumps are also called as H plus ATPase pump. Okay, H plus ATPase found within the membrane. So in this way they maintain the pH of the lysosome. Now most of the lysosomal membrane proteins, I have shown you the H plus ATPase or the proton pumps. Other proteins are also found. The other proteins are also found, like some transmembrane proteins are there which transport the digested products towards the cytosol. So different type of transmembrane proteins are found within the membranes of the lysosome, and the question arises that why these proteins are not degraded by the acid hydrolase found inside the lysosomes. It's because these transmembrane proteins, are highly glycosylated. They are modified with carbohydrate moieties, which carbohydrate molecules, which perhaps protects them from the lysosomal protease enzymes. So that's why the membrane proteins found within the lysosomes are not degraded because they are not degraded by the acid hydrolases found inside the lysosomes, because these proteins are highly glycosylated, the transmembrane proteins, like the proton pumps that we have said, then other transmembrane proteins which transport the final products of digestion into the cytosol. These transmembrane proteins are actually modified with, they are actually glycosylated, okay, by, they form a unique structure with the help of the carbohydrate, which helps them to protect from getting degraded by these lysosomal enzymes. Okay, now we will discuss about the types of lysosome. So let us discuss the types of lysosome now. Now there are different types of lysosomes depending on their processing, where they are getting processed actually. Like there is primary lysosome. I'm changing the color. There is primary lysosome. Primary lysosome, also known as the starch granule. Primary lysosome is also known as the starch granule. It is a small body having enzymes synthesized by the ribosomes, which are attached to the endoplasmic reticulum. So simple thing, primary ribosome are actually called as starch granule. They are also known as starch granule, and they are actually synthesized by the rough endoplasmic reticulum. The enzymes which are found within the primary lysosome, these enzymes are actually synthesized from the rough endoplasmic reticulum. They are, the ribosomes are attached with the endoplasmic reticulum, and after synthesizing that lysosomal enzymes, the primary lysosomal enzymes, that enzymes enter to the lumen of the endoplasmic reticulum. From there, it goes to the Golgi complex. Now, inside the Golgi complex, an acid phosphatase reaction takes place before they are transported to the primary lysosome. So, from the endoplasmic reticulum, they are transported to the Golgi apparatus, and within the Golgi apparatus, what happened? An acid phosphatase reaction takes place. An acid phosphatase reaction takes place within the of the enzymes which will be found in the primary lysosome. Okay, and then they will be transported to the lysosomes, or what we call as primary lysosomes. Okay, so primary lysosomes, also called as starch granule, they are primary lysosomes are small bodies. They also have enzymes, and that enzymes are actually first synthesized by the rough endoplasmic reticulum, better to say. Rough endoplasmic reticulum have ribosomes attached to the membranes of the endoplasmic reticulum, where they translate, they synthesize the enzymes needed for the primary lysosome formation. Then they will enter to the Golgi complex, where the acid phosphatase reaction will takes place. Before they will be transporting to the primary lysosome, within the Golgi complex, acid phosphatase reaction will take place, which may be preferentially charged with one type of enzyme or another. Okay, so this was about the primary lysosome. Now you should remember that acid hydrolase are found on the secondary lysosome, right? Okay, so now we will discuss about the secondary lysosome in detail. So primary lysosome, I have said, just simply you have to learn that they are known as starch granule. The enzymes of the primary lysosomes are synthesized by the rough endoplasmic reticulum. Then they are transported to the Golgi apparatus, where they are further reaction occurs. Acid phosphatase reaction takes place, and which during the acid phosphatase reaction, these enzymes are preferentially charged with one type of enzyme or another, and then they are transported to the primary lysosome. So one type of enzyme will be present. Then the secondary lysosome secondary lysosome, also known as heterophagosome or digestive vacuole, and secondary lysosome mainly occurs due to your phagocytosis. It occurs due to phagocytosis or pinocytosis. You have learned this term when I have talked to you during the plasma membrane, right? Pinocytosis. So during the time of phagocytosis or pinocytosis of a foreign material by the cell, secondary lysosome observe. And secondary lysosome, also known as what? Heterophagosome. It is also known as heterophagosome, or also known as digestive vacuole. Secondary lysosome, also called as digestive vacuole. Let me write in a good way. So secondary lysosome, I'm writing in small suffix sl. It is also known as heterophagosome, also called as digestive vacuole. Now, secondary lysosome occurs where due to phagocytosis or pinocytosis by a foreign material. Okay, so when the foreign material undergoes phagocytosis or pinocytosis by the cell, then secondary lysosome observe. It shows positive phosphatase reaction which may be due to association of so secondary lysosome actually shows positive phosphatase reaction which may be due to association with the primary lysosome. So secondary lysosome or heterophagosome, they associate with the primary lysosome. Okay, and that's why it shows a positive phosphatase reaction. The engulf material, that means the material which is inserted, the foreign material which is introduced by the cell through the process of phagocytosis or pinocytosis, that material will be digested by the hydrolytic enzymes present on the secondary lysosome, and the products of the digestion through the membrane of lysosome are will be incorporated to the cell. Okay, so that's the transmembrane proteins that I have said. They will after digestion of the materials, the products of the digestion will be transported out from the lysosome by the help of transmembrane proteins found within the lysosome, secondary lysosome here. Okay, so this was about the secondary lysosome. So secondary lysosome, also known as heterophagosome or digestive vacuole, and when secondary lysosomes are observed, secondary lysosomes are observed during the process of phagocytosis. They occur during the process of phagocytosis or when we see pinocytosis. Both of these process actually what they do during these two process, your foreign material are introduced by the cell by these two process. There is also receptor mediated endocytosis which we have done before. It is mediated by the clathrin proteins, right? And this secondary lysosome associate with the primary lysosome, and the engulfed material, that is the material which is introduced by the cell, that material will be digested by the hydrolytic enzymes present on this secondary lysosome, and the products of these digestive products, the products of the digestion, now they will be transported through the help of transmembrane proteins found within the membranes of the secondary lysosome out from the lysosome to the cytosol, where that digested products will be excreted out from the cell, or they will be reutilized by the cell. So I hope you have understood all. Okay, now we will discuss three more important thing, that is residual bodies. Residual bodies. So residual bodies are those bodies which are formed if the digestion is incomplete. So incomplete digestion leads to the formation of residual bodies. Normally these residual bodies can be removed by a process called defecation. Defecation. So by the process of defecation, residual bodies can be removed by some protozoa. So some protozoa have ability to remove these residual bodies by a process called defecation, but others, these residual bodies will remain within the cell, and that's why it plays an important role in the aging process, cell aging. So residual bodies are what? They are incomplete digestion by the lysosomal enzymes that leads to the formation of residual bodies. These residual bodies can be removed by a process called defecation in some protozoa, but other cells may cannot able to remove these residual bodies and they may persist for a long time, and that's why these residual bodies leads to aging of the cell. It can leads to aging of the cell. Okay, incomplete digestion due to the absence of some enzymes. Now, why incomplete digestion occurs? That's the main question. Incomplete digestion of the lysosomes occur due to the absence of some enzymes within the lysosomes that leads to the formation of residual bodies, and it also leads to the accumulation of phospholipids, sphingolipids in some cases, and sometimes glycogen in some other cases. This leads to severe pathologic disturbance. So residual bodies are result as residual bodies occur as a result of incomplete digestion. These residual bodies occur as a result of incomplete digestion. Now, why incomplete digestion occur? Simply incomplete digestion occur due to absence of absence of some enzymes. Some enzymes found within the lysosomes. Okay, okay, and they remain within the cell, and due to which what happen, the phospholipids, the sphingolipids in some case, or glycogen may remain within the cell, and as a result it can lead to severe pathologic disturbance. It also leads to aging of the cell. So I hope you have understood about the residual bodies. Generally, residual bodies can be removed by defecation process by some protozoa, but others, they accumulate within the cell, and that's why it leads to aging of the cell. It leads to some severe pathologic disturbance within the cell, because undigested part remain in the cell, they are not utilized, neither they are excreted out, they remain within the cell. In this way, if one after another, if residual bodies get accumulated, then it will disturb the normal function of the cell, obviously. Next, we will say about the autophagic vacuole. This is more dangerous. Autophagic vacuole occurs sometimes, that we know. Okay, so let us write about autophagic vacuole. Autophagic vacuole. I am repeatedly writing wrong spelling. Vacuole, VCUOLE. Now, autophagic vacuole, also known as autophagosome, it is also known as autophagosome. Now, this autophagosome is a lysosome which contains a part of a cell itself for digestion. That means autophagic vacuole are those lysosomes which actually its digest the cell's parts. Own parts are getting digested by the lysosome itself. So intracellularly they are digesting. They are digesting the interior parts of the cell. Okay, now these interior parts involved in digestion may include some parts of mitochondria, some parts of endoplasmic reticulum, and they are formed during physiologic and pathologic process. So autophagosome, they are actually what they are digesting? They are digesting the intracellular parts. They are digesting the intracellular parts. That means the parts which are found within the cell, like mitochondria, endoplasmic reticulum, right? And that leads to pathologic conditions. It leads to death of the cell. So under certain condition, it has been shown that autophagic vacuole may cause digestion without irreparable damage. That means they will not repair, they will not reverse the damage. Okay, the damage that caused by autophagosome is irreversible because they are digesting their own intracellular parts, right? So it is irreversible, that is done by autophagosome. So autophagosome or autophagic vacuole are those lysosome which digest the intracellular parts of the cell itself for digestion and which cause irreversible damage. That means they will not able to repair the damage. It leads to death of the cell. The last type of vacuole are the last type of lysosome is your plant and fungal vacuole. Let us see. Well, plant and fungal vacuole, plant and fungal vacuole, plant and fungal vacuole occupies almostly 30 to 90% of the plant cells, and the plant and fungal vacuole can be comparable with the lysosome of the animal cell. Plant and fungal vacuole also contain the enzymes. They also contain the hydrolytic enzymes, but they also perform other functions as well. So plant and fungal vacuole also contain hydrolytic enzymes. Okay, they also contain hydrolytic enzymes. That's why we are comparing plant and fungal vacuole with animal lysosome. But the function of plant and fungal vacuole not only digest, but they also have other functions as well. What are the other functions? Like the first function is storage of nutrients and waste products. So the first function is storage. Okay, storage of nutrients and waste products. Okay, storage of nutrients and waste products like your proteins, like your some plants, they store anthocyanin, rubber, etc. Okay, second function of this type of lysosome found in animal and fungal cell are degradation of the waste material. That is also done by our animal cells also. Degradation of waste material, then increase of cell size, obviously increase of cell size and control of turgor pressure. They also maintain the turgor pressure. Different type of pressures are maintained by the cells to maintain the cell structure of plant cell. Turgor pressure is one of them, right? So different functions performed by the vacuole found within the plant and animal cells, plant and fungal cells. Within this vacuole, plant and animal cells store the hydrolytic enzymes for digestion. At the same time, they store the nutrients and waste products. They degrade the waste products, increase the cell size, and control the turgor pressure. Now, this vacuole found within the plant cell and animal plant cell and fungal cell, they also actually maintain the pH also. Okay, uh, like for instance, if there is drop of the pH, suppose the pH level falls down in within the environment, then the flux of H plus ion into the cytosol will be balanced by corresponding transport of the H plus ion into the vacuole. So they will maintain the pH inside the cell. Okay, if there is change in the pH outside the environment, then they will balance that pH by transporting the H plus ion into the vacuole, into the lysosome, what we call as plant and fungal vacuole here, and thus it will maintain the pH in the cytosol constant. Similarly, the turgor pressure, as I have said, the turgor pressure is also maintained by these plant vacuole, plant and fungal vacuole, which by and how they maintain the turgor pressure by transport of the sugars, by transport of the amino acids. If there is change in the osmotic pressure, pressure of the surrounding cell, then they will transport them, the sugars and amino acid, to maintain that turgor pressure. So this is about the your fungal and plant vacuole, which is similar with the animal lysosome that are found within the animal cells. So they perform a variety of function. They maintain the turgor pressure. How they maintain the turgor pressure? By transport of the sugars, by transport of the amino acid. Okay, within them to maintain the turgor pressure. Secondly, they also maintain the pH. They It is a type of homeostatic device. So your vacuole that we found, it is a type of homeostatic device. Okay, which maintains the pH. Suppose there is drop of pH outside the environment, they constantly allowing the H plus ion into the cytosol to maintain that same constant pH. At the same time, they also store the nutrients and waste products. They degrade the waste products due to the presence of hydrolytic enzymes. They increase the cell size, and I have said also they control the turgor pressure. So this was all about the structure and some of the functions of the lysosomes. In the next video I will discuss about the functions of the lysosome little bit in detail. Okay, so that's for all for today's lecture. If you have any doubts regarding this lecture, you can ask me in the WhatsApp group. Okay.